Well, oh my goodness, I can't be more excited for this talk. Um, just have been chomping at the bit to get to this. Emily Casanova is going to talk to us today about autism and evolution and human genetics from our side of things, which is almost never done. And there's such a prejudice against the primal, the archaic, our ancestry. I know this from working with great apes. There's a huge prejudice about identifying with anything ancient or that came before us, and I feel just the opposite. I love the idea of our ancestors and our common ancestors for sure, bringing us all together. It's a beautiful thing. Um, so points of order. We're gonna let Emily do her talk first, and then have a Q&A. If you want something read out loud or you have a question, just put read before you comment or add a question in the chat. We will get to it. Um, we're usually really great at being nice to each other and taking turns, so let's continue that tradition. And I don't want to waste any more time. Let's turn it over to Emily.
Righty. Well, thank you so much for having me, Dawn and Roger, and everyone else that's attending. I know Dawn said she was excited, and I was equally as excited because I was telling her that I've never gotten to meet her before, but I've known of her for good Lord, more than 2 decades now, and so that was really fun. That was a nice email to receive. So so that's great. So I'm going to kind of collapse my little window up here so I can actually see what I'm reading. So so I think Roger's going to try and help field if there's any comments or questions along the way, if anybody just has to ask something, which you know, as I said, we can save it towards the end, but if you really have something you got to get off your chest, that's okay. We can kind of roll with it. So but yeah, so my name is Dr. Emily Casanova. And I'm going to talk about some of my work today. I of course study primarily autism from a variety of different viewpoints. And today I'm going to talk a little bit more about my passion for evolution and kind of combining these two interesting areas, as well as human genetics. So let's gonna jump into it. There we go. Okay. So first, I feel like it's kind of probably helpful for you to know a little bit more about me and why in the world I'm even studying this stuff in the first place, and what is some more of my personal background. And so, of course, I am a scientist, as well as a teacher, and I study autism primarily. I do a lot of overlap with autism and from different evolutionary contexts. But I also study autism and Ehlers-Danlos syndrome. Some of you may already be familiar with me from that area. And so I'm also autistic. And then later on, I was diagnosed with generalized hypermobility spectrum disorder. So, and suddenly realized there's a lot of autism in that group. Hence, that's what got me into studying that area. So it's a little strange. I kind of have these two major viewpoints in terms of studying autism from evolutionary perspective, as well as more from like a clinical perspective. And so hoping eventually those can kind of merge again, but we're kind of waiting for some Ehlers-Danlos whole genome sequencing to be made available. And hopefully someday we can kind of start pulling that back in too. But basically, as far as my background from a personal perspective, I became part of the online autistic community probably around 2003-ish. And that was part of my own journey of initially self-diagnosis and then later on diagnosis. I was on Wrong Planet probably almost every day. And I was for a while there, I was a moderator on there, and eventually we kind of broke off and formed our own forum, which is sadly now defunct. It was called Gestalt, but it was a nice little autism family there that we had. But my avatar on Wrong Planet was often Mr. Bean, so that's why I put that up there in case that jogs anybody's memory. And my handle on there was Sophist. I was very full of myself back then. I was younger. So, but that was really the place where I first became familiar with the idea of the Neanderthal theory of autism. And of course, that was originally popularized by Leif Ekblad, he was known as Ardos on Wrong Planet, and he also was the developer of the Aspie quiz, which had a lot to do with some of his ideas on Neanderthal behavior and autism. So I was definitely by far not the first. I just do genetics work on it. So that's my claim to fame. He started the whole thing. As far as my professional background, it's a little bit of everything. My training is originally in neuroscience, technically in anatomy and neurobiology. And then in my postdoc, I got additional training in genetics and bioinformatics, as well as some clinical training to be able to do assessments for Ehlers-Danlos for research purposes. I'm not a clinician, so I can't diagnose anybody. But aside from that, I've really had a long time passion for studying different aspects of evolution and paleobiology. It's kind of been my nerdy side hobby for a long time, and I've really enjoyed reading all sorts of stuff from books about the Cambrian explosion, like Stephen Jay Gould's wonderful life book, all the way up to all sorts of human evolution books. And so it's really only been over the last 4 or 5 years that I've really been able to start combining this interest in paleobiology and evolution with some of my autism work. And it was really just happenstance because I was trying to study different different functional features and traits of autism related genes. I started noticing that they were somewhat predictable in terms of, for instance, their overall size. They tend to be ridiculously long, and they produce more complex proteins, which means they might be able to do a wider variety of things within the brain. And ultimately finding that a lot of these genes are really conserved. They don't like to change very much over time. So I'll talk a little bit more about that kind of towards the end. After I do all the Neanderthal stuff, I'll talk about a few of my other papers if that's of interest to anyone. So, but that kind of got me into it. And from there, I've just really been trying to merge my interest with autism and genetics and data science together. And so that's kind of led us into studying Neanderthal DNA and Denisovan DNA, which is thankfully freely available online. If you know how to, you can download it. And the Neanderthal one's a little bit more challenging, but Denisovan is easy enough. And we had access to my collaborator had access to an autism database. So it kind of started from there, and just a little honestly Hail Mary kind of shot in the dark pass, right? And it worked so far. So that was great. That was lucky. So as far as, just in case you're interested in seeing some more of my other stuff, where you can find me online, and granted, this is, Lord knows what's going to happen to TikTok in America, but anyways, if it still exists, you can find me on there. I do, as well as on YouTube, and I do a good number of videos. Some of them are on my research, but then more so, it's just trying to take a lot of autism related topics that I think people probably want to hear more of, and especially stuff that overlaps with like Ehlers-Danlos and related conditions like autonomic, dysautonomia and mast cell disorders and stuff like that. And just really try to kind provide a little bit more of the science in a, you know, kind of a more video friendly format, which so far people seem to like a lot. So I've been really happy with that. I also have blogged since 2012. My blog is Science Over a Cuppa. And although I've kind of been bad with that this last year since I started doing TikTok videos, I really need to get back to doing some of the blogging because I really do enjoy reading and writing, and it might be a little weird as far as this is the header on my blog, the fossil ichthyosaur, and it's just because I have a kind of a mini obsession with Mary Anning and her all of her wonderful finds, and including this ichthyosaur that was one of their first major finds, and she was like a young teenager when she found it. So anyways, so I just really she's somebody I admire. So hence the strange ichthyosaur on a sort of autism related blog. Why not? So okay, so that was about me. And if you guys have any questions, or you know didn't manage to get a screenshot, you know, definitely just you can email me. You can find me on Loyola University New Orleans website. I'm pretty much the only Casanova there. You can find my email if you want to get in touch with me. But anyways, so I just want to kind of give a little bit of background in terms of some of the basics of human evolution, so we can all kind of be on the same page when I get to the Neanderthal stuff. So just first off, and this is a little part of my lecture that I love to torture my students with in my brain evolution class. So so go ahead and get ready for a little ride. So first off, we've got basically the hominin lineage, the hominin family tree, fancy word for human. And the hominins first evolved in a period up here known as the late Miocene. So started 23 million years ago, roughly, and ran close to 5 million years. So we evolved a little closer to the more recent. So that's what they call the late Miocene. And so at least so far, the oldest potential hominin that's been found is about 7 million years old, and that's Sahelanthropus chadensis. I've probably mutilated that somewhat. But here's a reconstruction of our little Chad guy here. There are debates about some of these really old ones. So anything really over 5 million years old, the respective paleoanthropologists are still arguing about. So none of those are absolutely definitive. The only ones that are really undisputed are the Australopithecines, like Lucy. And these evolved during the Pliocene period. They lived roughly about 4 million to 1 million years ago. And of course, that meant that they were overlapping some species within our own genus known as Homo. So that actually wasn't all that long ago. The first of our genus, at least that we have found so far to date, who knows if that will change in future, but that was Homo habilis, and he was otherwise known as the handyman because he was the first one that they really found associated tools with. And he lived in Africa about a little over 2 million to about 1.5 million years ago. He overlapped with Homo erectus. Homo erectus evolved roughly about 2 million years ago, and then lived up until very recently, which is a mere 117,000 years ago, if not even a little bit more recently. So that is a drop in the bucket of evolutionary time. That actually is not that long ago. So. Okay, so the next one is Homo heidelbergensis / Homo antecessor. I'm throwing these two together because there is ongoing debate as to whether this was a single species or two separate species in different regions. So we're just going to throw them together. But this complicated species, lived in Eurasia and Africa from about 700,000 to 200,000 years ago. And this is our direct ancestor, as well as the direct ancestor of the Neanderthals. And so while they evolved from Homo heidelbergensis / antecessor up in Eurasia, we evolved from the same down in Africa. Of course, and then eventually a subset of Homo sapiens moved out of Africa into Eurasia, and that's how we ultimately met Neanderthals in the first place. So Neanderthals, they evolved probably roughly around 400,000 years ago at least, and seem to have died out by about 37,000 years ago. Not counting ourselves, of course, because we are technically hybrids, even though they are the minority species in that hybridization, but they are still technically alive, at least a little bit. But at least the full-blooded Neanderthals don't appear to have lived beyond that point. But who knows? It may well change again in future. But and then finally, Homo sapiens. We seem to have evolved at least as early as, maybe as early as 300,000 years ago in Africa. The oldest fossils have been found in a region of Morocco known as Jebel Erud, and that's in northwestern Africa. And so those are currently kind of the benchmark. These guys definitely have some transitional features that we see that are more modern human-like, but also a little bit more older or archaic. So they definitely have features of both. But this is the one where we can kind of argue this is probably one of the earliest ones that seem to be roughly Homo sapiens. So again, species is a semantic concept. So and paleoanthropologists love to argue about that stuff. So anyways, we'll leave it to them. So, okay, so the hominid family tree, just to kind of see it all together, we've got the Ardipithecines, which are some of the older ones. Those are the ones that are still kind of under debate. And then we've got two groups of Australopithecines. We've got the robust ones and also the gracile ones. Lucy was one of the gracile ones. And then finally, we've got our genus Homo up here, right? So this is a very clean version. And speaking of clean, I want to talk a little bit about how we tend to view speciation, which is an important topic when it comes to then what does intermixing actually mean and create, right? And so typically in evolutionary biology, scientists prefer to think of life and speciation as this classic bushy tree. This is very much like Darwin's original drawings, right? But in actuality, because life is typically messy, speciation is usually not as clean as that. And so instead, rather than a bushy tree, it usually tends to look a little bit more like a braided stream. And that is more of the predominant metaphor that's probably a little bit more accurate. And that's definitely something that describes our family tree, at least in the more recent past, and I'm sure even more ancient past than that. So, you know, it's hard to catch hybridization in and intermixing in progress. And it doesn't always show up very easily in the fossil record. And so we've probably consistently underestimated how often this happens, especially between closely related species or subspecies. So there's definitely been a lot of intermixing between different species of Homo. And so the ones that we're really clear on, of course, are the Homo sapiens, ourselves, the Neanderthals, and the Denisovans. The Denisovans were out in Asia. We tended to think that most of this intermixing happened like the last 100,000 years, but really that was just the most recent round of intermixing. And so that has actually happened for at least a couple 100,000 years, if not even more. Now, I will say that the stuff that we're going to be talking about today is strictly the result of the most recent intermixing, roughly, 40, 50-ish thousand years ago. And the reason being is that none of the previous intermixings have any offspring that are still alive to date. So Homo sapiens that had intermixed with Neanderthals, for instance, like 200 plus thousand years ago, we see that in the DNA of Neanderthals, but those humans, those Homo sapiens are no longer alive. They didn't have any progeny that is still alive. So we don't see the back mix in that. So we only really see one kind of protracted round of intermixing in modern human DNA, which probably happened for a couple thousand years, most likely around the Levant region, which is really where most likely Homo sapiens coming out of Africa were really first meeting up with the Neanderthals that time, and probably living in and amongst and in close relation with these Neanderthals. So definitely a lot of intermixing, and we also see evidence nowadays in Asian and Native American genomes of intermixing with Denisovans from multiple different rounds. That was strictly in modern day Asians, and of course Native Americans are an ancient subset of Asians, so they also, and they broke off after they had intermixed with Denisovans, so we see Denisovan DNA in people who have Native American ancestry too, which includes a lot of Hispanic people from the Americas. So it's a really interesting way to track people over really deep time too, not just to study things like autism and other neurodivergent conditions. So anyways, I like this for a lot of different reasons. Hopefully you're finding this interesting as well. So getting to the meat of it, why autism and Neanderthal DNA? As I said, I'm definitely not the first person to think about this relationship in terms of autism behavior and possible links with Neanderthal behavior. Leif Ekblad was, I don't know if he was the first, but he was certainly the first to write about it and popularize it. And that was definitely a more popular idea back like 20 years ago, and Wrong Planet and some of the others. It was definitely a popular conversation, right? And I haven't heard as much of it as late, but hopefully maybe with some of this, it's coming back into conversation a little bit because I think it's interesting. I think some of the reasons why it has kind of had multiple rounds of kind of popularity is because it does, the concept is kind of appealing, I think, to a lot of folks in the autism community. For the same reasons, I think probably the alien was a popular logo for Wrong Planet. And it really just kind of speaks to this sense of otherness that we often experience growing up and feeling like we don't always fit very well into, you know, broader culture, right? And so alien is very easy to understand, but I think even feeling like a different species like Neanderthal, that also has kind of broad appeal for our community. So I think that's probably why it's kind of hung on a bit. Now, I will say as a scientist that just because something is culturally appealing doesn't necessarily make it true. And so I will say that I found it interesting, but I didn't really find it terribly convincing at the time, mainly because I wasn't as interested in the behavioral components so much as more the biology, but that's my personal preference because that's where I come from. So it really wasn't until a number of years later, I was reading Simon Baron-Cohen's book, The Pattern Seekers, and he was talking in one of his chapters about something in ancient human history I hadn't heard of until that point, because I am not an anthropologist in the classic sense, or an archaeologist, and that is the Upper Paleolithic Revolution, and that began about roughly 50,000 years ago. And basically what it refers to is a major change in the ways that our species, particularly within Eurasia, primarily because Africa does not have a very good archaeological record, unfortunately, because of the peculiarities of the landscape. And also too, not as many people have been studying in Africa too. So I'm hoping that will change over time. I think it is slowly changing, but basically it's the way that these people, it's a major change in the way that they made tools, the way that they traveled, the way that they traded goods, and basically produced materials, even the way that they basically kind of invented rooms within their areas in which they were living, which really wasn't apparent before. And so all of a sudden there was a room, there was an area for sleeping, there was an area for toiletry, there was an area for cooking, there was an area for preparing the meat, all of these different kind of breakdowns of the space, which wasn't apparent prior. either in our species or any other of the human species. So this was something that was kind of striking to the archaeologists and anthropologists who were studying this time period. So I found it immediately interesting because I kind of knew the rough time period when we were supposed to have intermixed with Neanderthals. And it was roughly around that time period. And I thought to myself that that sounded like a great big coincidence. Maybe it wasn't a coincidence after all. And the other reason was because I was already very familiar with a lot of literature on other species in terms of the biology of hybrids and what that often can mean in a deep time perspective. Hybridization is often a stimulus for kind of new evolutionary change, partly because smushing two species together that have not been together for some time can be a destabilizing event from a biological perspective, which is really interesting. It can bring up some nasty stuff, right? So there probably, for instance, was a couple thousand years where we were intermixing with Neanderthals, where there was probably much higher rates of fertility problems, probably much higher rates of congenital problems, infants that were, being born with a lot of physical problems. That probably took time to kind of weed some of that out for a while, stuff that may have been deadly, right? But on the other hand, it's also an opportunity for some new interesting change. And that's the kind of thing that I like. And so it did make me wonder whether, if we saw this, this upper Paleolithic revolution right around that same time, or kind of starting from that same time, did these hybridizations, did they play some role in kind of stimulating new things that weren't necessarily just because we were Homo sapiens and we were going to develop that way anyways? Almost typically it's tended to have been treated as though this was an inevitability. We were just that smart and we were going to, outcompete Neanderthals, and there you go, right? Because it was always a perspective in terms of competition, very Darwinian in that sense. But there's me thinking, was this actually a stimulus for some of this cognitive change? Because again, we are hybrids. We are not purely Homo sapien, at least not what we were prior to that intermixing. And then there's also some of these links, of course, having come from the autism research community and the autism community in general, a lot of these links between autism and savantism and creativity and intelligence. And yeah, I know there's a lot of challenges too, but these are some of the things that, can be really kind of cool, right? And so again, it was like, well, are some of these popping up because maybe we got a little bit more, or certain types of Neanderthal DNA that in some ways, maybe it's not always working so well. There can be a little bit of, kind of genetic mismatch, which can cause some problems. But on the other hand, you might also see some beneficial stuff popping up too at the same time. Stuff doesn't always have to be completely bad or completely good, right? That's a very human concept anyways. It's just all biology at the end of the day, right? The main thing is that ideally you have progeny and you pass it on. That's ultimately what evolution cares about, right? So this kind of leads to one big question. Again, you're probably already there because I've been saying it. Did this intermixing was this kind of a stimulus for our species? And some of the stuff that we're seeing in the Upper Paleolithic Revolution. I'm definitely not the first person to ask that. There was another group of anthropologists and archaeologists back in 2019 who proposed exactly that, although not from a biological perspective. It was more from a cultural connectivity perspective, in terms of kind of being a synergistic influence when two species come together and they share ideas and they share cultures and kind of become something new, right? Which there's probably definitely some truth to that as well. We certainly traded information, not just, beads. So, and parasites. We certainly traded parasites too. So, But from my perspective, you know, being an autism researcher, my question was, does Neanderthal DNA in modern populations now, does that somehow influence autism? And so that was kind of the big question. Again, it was a big Hail Mary, could have worked, could have didn't, all depended on what was there, right? And so I took my crazy idea, and I brought that to my one of my main collaborators, Alex Feltus, and he's a biologist and a computational scientist over at Clemson University. And thankfully, he already had access to a subset of the Spark autism data set, which is one of the big genetics data sets. He didn't have whole genome sequences. He had exome sequences, which is like a smaller portion of the genome, but it's a good place to start. And he also had access to another database called GTEx, which we were able to use for controls, right? Because you always have to compare it to something. Now, I will say one of the things that's really important to do in these kind of genetic studies is that you really need to try to control for ethnicity, because of course, especially with any genetics, but we can especially see this as the case with Neanderthal DNA and Denisovan DNA in modern populations. Ethnicity plays a huge role. And so you want to make sure that you're actually measuring what you think you're measuring and not just differences between ethnic populations, right? I mean, if there's differences in autism susceptibility across different ethnic populations, that's interesting. That's one thing. But you don't want to think you're measuring autism and you're actually just measuring two differences between two different ethnic populations. So initially in the first paper, which was published last May, we had utilized self-reported ethnicities, and that's very common in these types of data sets. And we also took it a step further, and we looked not only at all of the Neanderthal DNA together, but we also further broke it down into common and rare DNA variants. A common variant is anything that's occurring in at least 1% of the general population. In this instance, we're having to use the specific matched ethnicity as a guide. And then rare is anything that's under 1%. And so it's a good way to see, you know, you can almost infer some of the functionality based on how common something is versus how rare it is as well. So for each individual in these data sets, we assign them 3 major scores. The first one was just a percentage of, you know, how many Neanderthal SNPs did they have, right? How many were they carrying? And we just gave them an overall percentage score. And this has been used in other research studies. We didn't invent this approach. But then we divided that then into the common ones and then the rare ones. And we were able to look at these each individually. The reason that we were so interested in particular in the rare Neander scores is, for one, like I said, I talked about, you know, not everything in biology has to necessarily be good or bad, right? It can be a mix of things. Sometimes there can be benefits to a particular DNA variant under certain contexts, and sometimes it can be a bit of a detriment. And so it can really vary. And so the main thing with autism is, of course, on average, while there are definitely plenty of autistic people who have kids, on average, we don't have kids at quite as higher rates as the general population does, right? And so that means that regardless of what those DNA variants that are doing, the stuff that's more related to autism is a little bit less likely to get passed down, just simply because we're just not having kids at quite the same rates, right? So that's why we're expecting to see maybe some of these rare Neanderthal variants have been affected by that, right? They're becoming a little bit more rare simply because they're just not getting passed down as often, right? Sometimes it's just sheer dumb luck, and it does have to do with ethnicity, and stuff does get weeded out just by sheer probability. But we're more interested in what maybe is the function of some of these. And so so the rare ones you can kind of see over here. I think I've got, there we go. There's my error as my arrow. So basically we looked at these rare Neander scores. The green ones are the black non-Hispanic people. The red is the white Hispanic, and then the blue are the white non-Hispanic. Those were the major groups that we had to work with. There were some other individuals who didn't quite fit in those, and we had to unfortunately kind of hold them off simply because there weren't large enough numbers to compare them with. So, but this was as simplified as we could get it. Now I will say that the lighter colors are the autism ones, the darker colors are the controls, the non-autistic controls. And you can see that consistently the autism group is exhibiting a lot more rare Neanderthal variants than their ethnically matched controls. Now I will say the white Hispanic group, take that with a grain of salt. The difference will not be that large simply because our control group, unfortunately, in GTEx was very paltry, and we had to go to another database, which still didn't have a huge number of Hispanic people for comparison. And so there was under 100 individuals, which is why you either have something that's common or rare. And if it's rare, it doesn't exist at all in the control individuals in that group. So in reality, it would not be that dramatically different, although I suspect the trend would still hold. And I'll show you some of the other work that we've done to address this afterwards. So. We also looked at individual DNA variants, otherwise known as SNPs. And so, for example, there were some other ones that we were able to identify that seemed to have links with different subgroups in autism. One example was there was a SNP in the USP47 gene, and we found this in 80% of Hispanic autistic males who had epilepsy. And that was compared to only 15% of Hispanic controls, which is a very dramatic difference. And this was actually not a rare variant either. This was actually one of the common variants. As you can tell, it's in 15% of the controls. So this seems to probably have links, especially to some of the epilepsy. It has been linked with other epileptic conditions, not necessarily related to autism, but not this particular DNA variant. So this one was kind of new. And then we also found that there was a missense mutation that was derived from Neanderthal DNA in Black people in the COX-10 gene. This is a gene that's really important for inhibitory neurons in the brain. They help to kind of maintain the balance between excitation and inhibition. And in animal models where this gene is knocked out, now granted, that's going to be a massive effect, right? You knock out a gene, you're taking out the whole gene, which can be quite different from just a missense mutation, which is affecting one of the amino acids in the protein and may well affect how the gene is, how the protein is functioning, but probably is not going to have a severe effect on, as taking out the whole gene, like just knocking it out entirely. But in animal models with this COX-10 gene knocked out, they do have many features of this excitatory inhibitory imbalance, which of course has been linked with some aspects of autism in the past too. So we had a longer list of stuff, and you're welcome to go in and look in the article. It is freely available. But just know that some of these, this is kind of taking the tip of the iceberg here, just because of statistical analysis. It's the stuff that is really, really big effect, it's the easiest to see statistically, whereas stuff, the iceberg that's just under the water, you're not really able to get at that very easily without having huge numbers of people. And so that's always a challenge in terms of finding creative ways. And that's precisely why we decided to lump DNA together into these big groups like common SNPs, rare SNPs. That way we could kind of pool them together and more likely find an effect, even though it's harder to then go in and see which ones are really the biggest factors in those groups. So that's kind of the major science. It was a really promising early work. We were very excited about it, but at the same time, we knew that some of the major challenges, like one of the major vulnerabilities of the work, was the ethnicity issue again. And so just because self-reporting is not 100% accurate, and we are all relatively mixed, and it is very hard to label that kind of biology, right? And so one of the things in this next stage that we've begun working on is called an admixture analysis. And basically we've started on trying to work on the Hispanic subset, because we really didn't have a good way to identify how much Native American ancestry different groups, different Hispanic groups have. I mean, we know it varies. So we had access to people from Peru, DNA from people from Mexico, from Colombia, and Puerto Rico, and they really tend to span the range of Native American ancestry, with Peruvians tending to have the most, followed by Mexicans, and then on down. And so basically with this admixture analysis is we were able to identify a subset of Neanderthal DNA that we could use as kind of measuring sticks for an indicator of how much Native American ancestry a given person has. And so we were able to develop essentially a Native American score. So each person could have their own score, and then you can take that and use it to control for whatever you were looking at. In this instance, we tested it out again on the rare Neander scores. And so we took our Hispanic subset and we had our comparison group, and we assigned each individual a Native American score using this DNA. They basically get a percentage. And those who have a higher percentage, of course, have more Native American ancestry. And then we use it just to kind of control statistically for whatever we're looking at. And in this case, it's the rare Neander scores. And so so far, that's worked really well. So you can kind of see in this little violin plot here to the right that autism still has much higher rare Neander scores compared to the controls, even after we have controlled for the Native American ancestry. We were especially trying to control for the Native American ancestry because Asian people tend to have the most Neanderthal variants, even more than European people. And as a subset of ancient Asians, Native Americans also have quite high Neanderthal DNA. And so basically we're looking at how much Native American ancestry versus how much European ancestry a given individual may have. Those tend to be the two major groups, especially in those regions that we had access to. So this is kind of, working case in point, seems to be working well. And so it's very gratifying to see that in spite of some of the earlier problems with the self-reported labels, we're still seeing the same effect, which is very heartening. And we're also going to be hopefully expanding to look at whole genome sequences, not just the exome that we had access to. So we're hoping to be able to identify a whole bunch of other DNA variants that may be influencing autism somehow. But we have other admixture analysis that we need to do. And so we need to do the same thing basically for African ancestry, Asian ancestry, and European ancestry. Those are the big groups that we need to control for. And so we'll be applying for grants. I don't know how that's going to go. I don't know if you all have heard about the whole NIH NSF debacle in the US. So we'll see if that exists in future. But ideally, we'd like to get funding for this to be able to hopefully get some more folks working with us and get some more space, because obviously whole genome sequences take up a lot of space. So that's the major stuff for this kind of series of work. If you're interested, we did publish a feature article in Scientific American on this. This was just back in February. So if you're interested, we talked about the autism stuff. We also talked about a little bit more general cognition, which I'm going to talk more about in the coming slides after this. If you want to go back and read it, I know Scientific American, if you get a subscription, they have their app and you can do back issues and stuff. Or I think Amazon also sells back issues and things like that. Or you can get it from your library, hopefully, which is good. Support your library. From that article, I want to talk a little bit about not just autism, but some other areas in terms of neurodivergence and just general cognition that Neanderthal DNA may be influencing. So there's been a number of studies before our study. We're just kind of the latest in a long string of very interesting work. But previous studies have found, for instance, that there's been some differences in relation to Neanderthal DNA and the shape of human skulls. So if we look at the differences between the classic Homo sapien skull versus the Neanderthal skull, our skulls are much more globular and the Neanderthal skulls are much more elongated. I always like to do this overlay for anybody who likes sports or football, any football. And we're more of a soccer ball, and Neanderthals are a little bit more of a American football shape. But there was a study that found that there were links between the amount of Neanderthal DNA that an individual has, especially they were studying people of European descent. And they found that the shape of their skulls differed the more that they had this Neanderthal DNA. In particular, they found that the back regions of the skull, around the occipital region and the parietal regions, were most affected, and they were somewhat reminiscent of the kind of a little bit more of the American football shape that you would expect to see. Not so much that you could just stare at the person and realize that they have a football shaped head. That's not what I'm saying. They had to do this using, using imaging, but the effect was certainly real. And that same group went on to do another study a little bit later, and they found that individuals who have, variations in Neander scores, so how much Neanderthal DNA you have, seem to have some of the biggest effects in a region known as the intraparietal sulcus within the brain. And in particular, there's a group of tracks of connections that are running underneath that region. And those seem to vary significantly. And so people who have higher Neander scores, they were much more likely to have an increase in connectivity in tracks through the IPS that were associated with visual processing. And you can see that on the picture in orange. Whereas those same people were more likely to have a underconnectivity or a decrease in connectivity in IPS connections that were associated with attention and social ability. And so I will say that aside from the Pattern Seekers book, this article in particular also really inspired my ideas about our series of studies because of the dichotomy that we often see between visual processing and social ability that is somewhat reminiscent of autism. And I'm not saying that everybody in the spectrum is phenomenal with visual processing. Obviously, it's a spectrum, but there is definitely a increased ability in terms of different aspects of visual processing that we do see in the spectrum more frequently. And obviously, social ability is not exactly our forte. So that's something that I think, probably many of you, if you've been around a lot of folks on the spectrum and have had an opportunity to be part of those communities, those kind of themes have probably popped up in one way or another many times. Obviously the social stuff, but the visual processing, right? How many times, did we talk about, Gestalt processing versus being able to, find that little puzzle piece and put things together without even having to look at the puzzle picture, right? You just hear a lot of those things. So this was definitely another work that strongly influenced my ideas and helped to give me that idea to try and look at autism and Neanderthal DNA in the first place. There are definitely, besides autism, there are definitely other associations that have been found so far. This is not something that my group has done. These are other investigators, but Neanderthal DNA in one way, shape, or form or another has been linked with major depression, with substance abuse propensity, with pain sensitivity, which probably I know a lot of folks who are thinking Ehlers-Danlos, aha, you know, some of the pain stuff is probably piquing your attention. As well as, of course, a greater consumption of pain-related medications, which goes along with the pain sensitivity thing, and ADHD. So all of these so far have been linked with Neanderthal DNA in various ways, not necessarily in the broad way that we did. Sometimes it's more about specific regions of the DNA or specific SNPs even, but there have definitely been links. And I suspect probably that list will just continue to grow over time. I will be surprised if almost anything is not linked with Neanderthal DNA in one way or another. And of course, it's not the only thing that you know, is interesting in the genetics in reference to autism. It's not, you know, like the sole root of autism. Autism is super complicated, but this is a really interesting viewpoint, and one of the things that does seem to be playing a role. And in future, our group is really hoping to expand to study Denisovan DNA as well, and in certain Asian and Native American populations. So do stay tuned for that research, as well as all this other stuff, which I imagine is just going to continue to probably speed up, and we'll find more and more links with various things. So that was my major Neanderthal DNA autism spiel. And as I said, I wanted to take a few minutes to talk about introduce you to some of my other areas of work. I already talked a little bit about the kind of ancient autism gene evolution thing. And one of the things that I was able to identify was basically that there are certain periods of evolution when certain gene groups have radiated or have evolved. A lot of autism genes seem to have evolved during the Cambrian period, during the Cambrian explosion, in the vertebrate lineage. So the earliest fish, right? And the really interesting thing about that is that's the first time we see basically the blueprint for the vertebrate brain. Before that point, animals definitely have a central nervous system, but not what biologists would technically call a brain. They have groups of neurons, but not what we would call a true three-part brain. And so this is the first time that evolved in the vertebrate lineage, and a whole chunk of autism genes seem to have evolved around that time. I know a lot of folks have found that strange in terms of why did so many genes evolve so quickly and all at once. And it literally was almost all at once, in part because it was the result of two separate whole genome duplications, which were retained in our lineage. And we still can see the remnants of those whole genome duplications, even in modern human DNA. That may sound strange because you're like, well, if it's duplicated the whole genome, why are autism genes special? Wouldn't that refer to all genes? Well, the interesting thing is that no, actually it doesn't. All the genes were definitely duplicated, but over time, a lot of genes are lost simply because extra copies are unnecessary. And so to lose them is no big loss. It's not a big deal. The exceptions tend to be with autism genes, other nervous system related genes, and other developmental genes. Anything that's really important for the development of the physical form, not so much the metabolism and the day-to-day workings, but how that blueprint is laid out, right? Those you cannot lose, usually without killing the animal, because they actually form these tight networks. And What's most important is the relative ratio of these different proteins in that network that work together rather than the total amount. And so when you duplicate that network, you cannot lose one player from that network without basically crashing the whole thing. So that is why these types of genes, autism genes are a prime example of this, but we see the same trend in nervous system and developmental genes. Only autism genes are on the really extreme end. And I'm really super fascinated by that because it's not just because they're nervous system or developmental. They're kind of their own thing in and of themselves too. So anyways, that's why a huge number of these evolved right around that time. And they probably had some really important roles in helping to lay down the vertebrate blueprint for the brain as at least the fish brain as we know it, and creating new regions and probably new neuron types. And because one neuron looks like another neuron under the microscope doesn't mean that in terms of identity, they're the same thing. There's a wide variety of different types of neurons. They all really know where they are in your brain, and they all have kind of their own unique identity. So anyways, so a lot of that complexity probably came out of genes like these autism related genes. So I'm continuing to work on this. I'm studying these groups of genes and their conservation patterns, how they like to mutate over time. They seem to like to be still for a really long time and then suddenly have a rapid pace of mutation and then kind of calm down again. So that's something kind of peculiar to them. So that's one area. And I will say I have a popular TikTok video on the Cambrian explosion of autism genes that people really seem to like. I didn't know how popular fish were in the autistic community. I probably should have known that, but that's why that totally went viral. Apparently that and Cambrian explosion. There's my other Cambrian explosion obsessed people too. So if you're interested in that, look me up on YouTube or on TikTok, and I've got a series of 3 videos on that, kind of exploring that. Another area of research that we're kind of wrapping up the first phase, and right now we've got a paper under review, is I'm studying Ehlers-Danlos syndrome, but right now I am studying it in women with Fragile X premutation, which is, of course, definitely has many links with autism, along with Fragile X syndrome. And so we have found that a subset of women with Fragile X premutation seem to have Ehlers-Danlos syndrome, which means that probably there is a subset of women with Ehlers-Danlos who are undiagnosed Fragile X premutation, simply because the majority of people with premutations are undiagnosed. And unless they have a kid with Fragile X syndrome, they're probably not going to find out. So that is a project down the line we would like to do. But anyways, we're finding that women who have this, who are premutation carriers and who have this Ehlers-Danlos-like phenotype seems to be hovering around there's a lower repeat number. They have a peculiar kind of genetics in the FMR1 gene. In the promoter of the gene, there's a whole string of repeats of CGG, CGG, CGG over and over and over again. Most individuals have anywhere between 26 and 32 of these, but premutation carriers, usually 50, 55 and over. Up to 200, that's considered the premutation, and then anything over 200 is considered full-blown Fragile X syndrome. But we are finding that women who are kind of on the lower end of that repeat, so 90 repeats and under, these are the ones who are much more likely to have Ehlers-Danlos syndrome. And so we talk in the paper, we will be talking in the paper once it gets published about potential mechanisms. And I strongly suspect that there are aspects of it that are I hate to say degenerative in nature, because of what we see with other Fragile X premutation related conditions that occur in that same repeat range. And we know, for instance, that same repeat range is associated with Fragile X associated premature ovarian insufficiency, so very, very, very early menopause. And we know that that has a degenerative mechanism. And so it's suggestive that probably the Ehlers-Danlos version in Fragile X premutation is a degenerative thing. Not rapidly, not like, Alzheimer's kind of degenerative, but certainly degenerative. And it has implications also for Ehlers-Danlos in general. I think there may be a subset that very well may be degenerative, and probably some of the links that we're seeing with inflammation and mitochondrial problems are probably speaking to that. So we'll see. It's early days. We still have to test that idea more to see if that holds water. But I suspect that's probably where that's going. This one is not really autism related, but I got into it thanks to autism. And I've been really interested in the theory of punctuated equilibria for some time. That, of course, was developed by Niles Eldredge and Stephen Jay Gould back in the early 1970s. And of course, it really rocked the evolutionary biology community because they were very used to this idea of Darwinian gradualism, where genetic change just happens slowly and cumulatively and bit by bit by bit by bit. And it's a very constant slow, gradual process. Whereas they proposed that change tends to happen more in punctuated bursts. And then in between, it's bookended by kind of these long periods of relative stasis where there may be a little bit of fluctuation, but there's nothing directional, right? Nothing that is really evolving. So I've had the opportunity to get to know Niles Eldredge. He's a good chum of mine, very sweet guy. I didn't know him before I wrote the original paper, and I sent it to him because I'm cheeky that way. And he was super nice. And even though he's not a genetics guy and that totally intimidates him, he was a very super nice guy. And he's been a nice mentor. Long distance, he's retired. He's up in Jersey, but a very nice guy. And I've been able to start working with some of his former students who are now full professors themselves. But it's been a lot of fun. And but anyways, we had developed this kind of a molecular explanation or a genetic explanation of punctuated equilibria. And we called it the developmental gene hypothesis. And this again is playing in with some of the autism stuff. The same mutation tendency, right? Autism genes don't like to evolve very quickly. They're very slow. They're very autistic in that way, right? They're very slow. They're very conserved. They don't like change. Same thing with a lot of nervous system and developmental genes. And so we kind of took this broader perspective that, of course, a lot of these genes are involved not just in brain and brain structure, but they're involved in body structure as well. And that's a lot of what you see represented in the fossil record. So a lot of these long, these starts and stops, I think the developmental genes are really playing a pretty big role in the patterns that are seen in the fossil record. So we've got a paper coming out, hopefully in a few weeks. It took a long time. Cambridge University Press unfortunately got hacked, and then it took months and months and months for them to get back to where they needed to be for editing. I did a lot of kind of revamping of the developmental gene hypothesis. This is kind of a continuation of that, as well as some mathematical modeling and some simulation studies predicting and basically applying a Nash equilibrium to explain the patterns of these developmental gene networks and how they don't like to change very much, but when they do start changing, then they change very rapidly, right? So it's very much a Nash equilibrium kind of thing. And then finally, because I just can't stop thinking and don't have enough to do, apparently, I have been working on an algorithm applying it to a GPT large language model to try and improve creative output, because that's always a constant problem for a lot of large language models like, Chat GPT and stuff. And basically taking some inspiration from some functional aspects of different neural systems, because of course I'm a neuroscientist, and trying to merge some of that. So I've been working on that. It's very early days, but I've got some promising output that hopefully might be useful in future, and improve creativity in some of these types of models. So that's kind of been the major stuff. So Right now I'm working at Loyola University in New Orleans, and up until the end of May, you're welcome to email me there if you feel like reaching out. But I will actually be joining the faculty at St. Louis University starting in June, and I'm going to be with the Department of Neurology as well as Psychiatry as an assistant professor and basic scientist. So I'll be able to do a lot more research, because right now I am teaching 3 classes in addition to doing all this work, and it is hard to find time for sleep sometimes. So I look forward to sleeping again, and but I still want to do some teaching. Definitely my brain evolution class is kind of my baby. I built that sucker from the ground up, and I would continue to want to use it. So hoping to be able to teach some classes there. So, but definitely do feel free to look me up. Please be patient with me. I am getting progressively busier and worse and worse with email. I am very nice and I try to respond to everybody. So you are more than welcome. If you've written me an email and I have not responded within a reasonable amount of time, do please feel free to nudge me. That will remind me. Because I'm just getting bad at juggling so many things. But I do enjoy interacting with our community, and I try to be an available resource. So so yeah, reach out, and if y'all have any questions, thank you so much again for the invitation. This has been fun, and I look forward to hopefully coming back and keeping in touch and stuff. So but yeah, if there's any questions, have at it.
Hey, Emily, I'm just going to jump in with a little summation of what's probably coming.
Sure.
As a cultural anthropologist, I loved during my work with apes and then other social animals, knowing for sure that I was experiencing a primal way of being, and that was at my core.
Yeah.
It's a beautiful thing. I love it. And one thing Casey has demonstrated is that cultural component, that way of being, some of us call it neuroholographic ways of being, are very connected and certainly not inferior in any way. In fact, often the opposite, very, very much connected, and
Right.
in a soulful way, if I may.
Mhm.
But we know from history and recent events that not everyone feels that way.
Yeah.
And so, of course, the worry, the fear among our members and broadly in the autistic community is the dangers associated with this kind of work. And I would love for you to really do a little bit of a deep dive into that.
Right, definitely. I will say, and I have been deep diving myself, and I have found that the main response that I have found most effective to people who bring that up is primarily because they are very ignorant of why we view Neanderthals and this caveman stereotype as so negative. And it actually gets people to shut up very quickly. And it is actually very founded and intermixed with racism. And I found that when I brought that up, and I say that very clearly, that those ideas that we have about what Neanderthals were like one, none of us has ever met a Neanderthal other than partly ourselves, right? So there's no way that we have any kind of real experience with them other than what we try to glean from the archaeological record and then try to glean from ourselves, right? So the concept of the troglodyte caveman is very intermixed and is very rooted in Victorian white armchair gentleman science. And so if you want to see the truth of that, you can go back and read, for instance, oh God, what was Huxley's book on he did one on anthropology, and there's sections. Now this was also at a time, keep in mind, where they did not know, they couldn't decide whether Neanderthals were a predecessor to human, and so they were just an older version of Homo sapien, or whether they were a different species. It really wasn't until after the 1900s when we had more complete reconstructions rather than just a skull cap and a brow ridge to be able to say this definitely seemed to be a different species. And so there was a whole lot of this that was intermixed with and around that time of savage cultures, and they were making similar comparisons. They were drawing the same comparisons, like within the same chapters between the brow ridge on a Neanderthal and the brow ridge on an Aboriginal Australian. And so this approach, this Victorian white male, affluent male approach, is very much rooted in racism. And I tend to bring that up, and usually people get very quiet pretty quickly, and they don't tend to follow that line of that accusation, right? Because I do know people, some people do not have, I think things are changing. I think in part because, of the, sequencing of the Neanderthal genome, the realization that we had intermixed, and so like attracts like to a certain extent. And then ironically, 23andMe has helped to popularize that. And so more and more people are seeing it in a positive way, but there are certainly remnants of that very negative stereotype of, of quote, primitive people. And we just need to not even have that conversation almost, you know what I mean? Like we need to call it what it is. And it really is a racist stereotype. Yeah, and that does get people to shut up pretty quickly nowadays. So, but I think it also helps to educate too at the same time, where they start to realize, oh, maybe that stereotype, maybe that wasn't as accurate as I thought it was, right?
And do you see any current dangers, since people aren't always rationally persuadable?
True. So far, I see a lot more positive stuff. I think that tide is turning. But I have had a few people approach me with those concerns, and I've told them, I've tried to tell them in a not a combative way, but what were the roots of that stereotype, and that it's not really reflective of who these people were, right? And we've really come at it and assumed that, they were almost subhuman. But then the Victorians did the same thing with anybody of color. So anybody who wasn't European, right? It was, they assumed the same. And there were even debates about whether, I mean, that was where the idea of the races came from that
Right.
Victorian time period.
Thank you. I'm sure other people might have
Yeah.
finer points to the question or whatever. And so I'll turn it over, but I wanted to say thank you in case my internet's been in and out. We've had really terrible storms. But yes, thank you so much. And I look forward to hearing the rest of the
Yeah.
conversation.
Definitely.
Great. So there is a question I'd like to read here. Says, can you say more about what you think the importance or advantages are of researching the genetics of autism, given the risks of using genetic data to select for non-autistic babies, for example, to prevent autistic babies from being born? And given that the autistic community has often claimed that hundreds of millions of dollars allocated to genetic research each year can be used instead to help autistics in their families, given high levels of unemployment and inadequacy of many existing services.
Yeah, those are all really good points. And and I definitely, even in my time back, 20 years ago, that was still a big concern in terms of how will this genetic research be used? Will it be used to benefit autistic people, or to lead to abortions, right? Because of course we saw, and we have seen that happen with Downs people. So first off, so far that has not really been happening, knock on wood. I will say as somebody who studies genetics, it is theoretically possible for people who have usually the more severe forms, the more severe expressions. Typically, a lot of the single gene hits or like a chromosome deletion or duplication or something along those lines, you almost always see some significant intellectual disability alongside the autism as well. Now, so far, with the exception of Downs, which definitely has a subset who are autistic, we have not seen that happen. Honestly, healthcare is so crap, they're not testing for anything. So, I mean, it is theoretically possible for a subset. Now, the minority of individuals, but I have not seen that happen because healthcare is just, in this instance, thankfully not good. And they're not testing for those kind of things. Now the ones where, like I said, they're these single genes, they have a very high likelihood of leading to intellectual disability and maybe autism. Those are potentially more at risk, but most individuals, in terms of their genetic background, it's freaking complicated, right? Like, I mean, even the Neanderthal stuff, right? We're talking about individual SNPs, which you're going to see in non-autistic people too, right? So it's not, these are not necessarily like really, really strong markers, right? It's not like Downs, right? It's not like you have an extra of chromosome 21, you're definitely going to get Downs, right? It's probably more like, hey, you have this, you have 15% more likelihood of being some kind of neurodivergent condition, depending on all these other factors together, right? So most of us have a very complicated genetic background. Same reason why we see a lot of our families, probably on both sides of our families, have many features, right? Because like attracts like. We're just throwing more and more of these genetic variants together, right? So most people are going to have these small factors that individually are not going to be good markers. They're not going to be useful for that kind of identification and abortion, right? So instead, and we're really, in terms of genetics, we're moving more towards, like, we've got the simple ones for the most part, the stuff that's like the big hits that cause the really, really severe stuff. We're moving towards more of the common variations, the ones that you got to pair with multiple different things in order to kind of reach cutoff to move towards autism, right? And so it really is kind of the human spectrum kind of thing, right? We're seeing us at the extreme end, genetically. And so most of us are going to have a very complex genetic background, that's going to be really hard to identify and not completely penetrant for autism. It's going to show up as different things for different people in different generations based on other factors, right? So they're not good markers, which is good in that sense. But on the other hand, it helps us to learn more about autism. What are the many different things that it is? Because everybody's flavor is different, even though we have many commonalities. We probably have more in common with autistic people within our families than, genetically and biologically, than people between, even though behaviorally and probably from a brain systems perspective, we probably have a lot in common still. But at that really fine level, at the genetic level, it's so varied. I mean, there's thousands of genes that have been identified as potential factors. That's a big portion of the human genome, actually. So yeah, so it's complicated. Now, in future, AI may be able to identify a lot of those, but it's still not going to be like a one-to-one thing where if you have this, you're going to have autism. So again, they're not going to be good markers. So.
Thank you for that detailed response.
Yeah.
It kind of makes me think, because there is a I guess there's fear of misuse and abuse of genetic data, and
Mm.
the testing, I guess the genetic testing is one theoretical use of or abuse of that data.
Mhm.
Do you know of some of the other risks that of genetics research that, I mean, as a person who's studying this,
Right, right.
that are concerning to you?
I mean, I would say just one thing, not maybe not so much a risk, but if you donate your DNA to a database that's going to be used for research, you don't exactly have approval over what types of research it's going to be used for. There may be stuff that you don't want it to be used for, or you're just not really interested in, or whatever. So I suppose that can be considered a risk because it's not being used for what you would like it to be, but it's kind of part of the agreement. So I do say, if you are going to donate, just make sure you've done your homework in terms of what range of things that database tends to be used for, which is sometimes hard to do. But if you're wondering, there's always people at those organizations you can reach out to and ask more questions to. Um, you know, that works as well. Like Spark. I've known people that are really interested in donating for Spark, but then others where they're concerned about some of the stuff we've talked about and they don't want to take part in it. And so it's everybody's a little bit different. And that's cool. I Spark data set, it can do a lot of good. It's probably not always been used for, I don't know, most stuff doesn't really go very far. A lot of research, getting it to a point that's actually useful and translational, that's like the hardest thing. And a lot of stuff doesn't pan out. That's just kind of the nature of research sometimes too.
Great. I think Don wanted me to read a comment. It says ties with creativity and intelligence. So any kind of attempts at, say, trying to prevent autistic bursts would be detrimental.
Yeah. I mean, it's so complicated. Like you say, I mean, yeah, are there things that we have challenges with, and everybody's going to vary somewhat? Certainly, but there's a lot of stuff that we can do. I don't know about you guys. I'm happy to be alive. So this is it's challenging, but there's a lot of cool stuff to do, and I want to do a lot of stuff before I kick the bucket, so I'm very glad that my parents decided to have me, and and I hope everybody gets that chance. Yeah, hearts definitely. So but yeah, there's a lot of, I mean, there's just so much creativity coming from our broad community. I mean, so many different ways, whether it's the arts, the sciences, inventions, engineering, etc. Even just day-to-day stuff, right? Like the simple stuff, because we always focus on, what are these big inventions? What did Mozart do? What did, Einstein, and those are wonderful, right? But even just the day-to-day stuff of, the autistic person who is a phenomenal gardener, and the little stuff that makes up day-to-day joy. And that is just as valuable, even if we were never even if we never produce Einsteins, right? Autistic people are still valuable because of our perspectives and because we're human beings, and we have those rights, and we're part of the broader human spectrum. It would be much shorter without us, and not as fun, I think.
Well, I think it's great that your work, you're able to use data sets that are already there.
Yeah.
So I guess the message is that the data is there, and it's data, and with data,
Yeah.
people can potentially use it to do a lot of harm,
Yeah.
but they can also use it to do a lot of good.
Yeah.
But if the data is not there, then you can't use it
Either.
to use good, right?
Yeah.
So is there enough protections in place that to prevent the harmful use of
I don't know about protections. I think right now we're just lucky that, like I said, healthcare is pretty lazy and cheap, and nobody's paying for that, right? It's hard enough to get a single genetic test done when you were an adult human being, let alone having a whole array of genetic tests done on an unborn child without, without justification because your insurance company is going to say no, right? That is kind of saving us right now. At some point, yeah, it is entirely possible that AI could make that a little bit more feasible. But I think, one of the things is that I've been doing a lot of reading on AI and a lot of the regulations that really need to come for it. And I think some of this can be cross-applied to that. Is we do need more regulations on that kind of stuff, right? If we are concerned about it happening, it's not so much about stopping all the research from happening, all the genetics research. Instead, it should be about trying to prevent its application in the ways that we are not okay with, right? And I think that's probably more fruitful and less about fighting the inevitable. Because honestly, I mean, even if I were to say like, yeah, no, that could totally happen and we don't need to do this, and I think we should stop genetics research, they're still going to do genetics research. They're still going to have genetics databases. It's still going to happen. And so I think at that point, we need to mitigate the risk in terms of, exactly like you're talking about, those have in place regulations that can help prevent that. That's, I think that's more assured. I think that's a better route to go, and to not stop the good stuff, right? Because I want to keep studying Neanderthal DNA and Denisovan DNA and all the cool stuff, and hopefully you guys like that and you want that to keep happening. But I need the Spark database in order to do that, because I don't have my own. I can't house over 100,000 people's genomes in my computer, even with a supercomputer. I don't have that massive a storage capability, and then all the lawyers that it takes to oversee how that data is used and doled out to different institutions. I mean, there's so many lawyers, you just wouldn't believe it. So there are a lot of regulations for certain kind of stuff, but in the traditional sense, universities are, big universities are still going to be able to have access to that ultimately, and to be able to use it. So yeah, so addressing regulations that you'd like to see at the application level probably would be more effective. And I think that would be feasible for people to come together from the neurodivergent communities and and try to talk with politicians and stuff, and get stuff in place. Because we have seen it in a very small form with Down syndrome, unfortunately. Which is a shame. I know folks with Downs can have a lot of health problems, and that's and especially like heart problems, and that's horrible, but they are such beautiful people. Every Downs person I've met is just like a light, and yeah, so anyways, yeah, soapbox, sorry.
No, sorry, I think I'm muted. Silent soapbox for me. No, thank you so much for that very insightful reply. We are coming to an hour and a half, so we do have to wrap it up. Just want to say thank you so very much for taking the time. And I think if anything, it's clear that we need more autistic genetics researchers,
Yeah, I would love to have more autistic genetics collaborators. So, or even other fields, right? I like cross-disciplinary stuff too.
Mm-hmm.
That's where you get the really cool inventions, when you're cross.
Oh yeah, I totally agree. I mean, it's about the different kinds of connectivity, and it's sort of human synergy at its best.
Yeah.
But
Yeah.
like, if you're willing, and I know Dawn would be super excited, we'd love to have you back, and
Yeah, totally.
hopefully
Yeah.
you know, we could maybe do some stuff together, you know,
That'd
with
be
regard
awesome.
to this.
That'd be awesome. Yeah, and like I said, I know our community likes a lot of Ehlers-Danlos stuff. So if y'all want anything on that too, more than happy to talk about that. So
Great.
cool.
Well, thanks again, and everyone, thank you for attending, and y'all have a good night.